Well, welcome to Chapter 7. Chapter 7, we're moving on to really something, there's still going to be a little bit of a philosophy, but as we saw even in Chapter 6 with Helmholtz, we've moved into an era that is beginning to look a lot more like experimental psychology. So even though with guys like Lotz and Helmholtz, they consider themselves to be physiologists, really, they're doing stuff that's very similar to psychology as we know it. And that's going to be the continuing trend here as we look into Chapter 7. We're going to see the emergence, also kind of separate a little bit from Helmholtz and somewhat not, of the study of the nervous system, right? Neuroscience. And again, that's not necessarily the same as experimental psychology, though from our perspective nowadays, so much of neuroscience does fall under the umbrella of psychology. Some of it does not. But also we're going to have a few other characters that show up here that are going to be much more obviously experimental psychologists, I think. So let's first deal with a little bit of philosophical background, just a little bit more here. Hobbes and Hartley are British materialists. Mind is motion, right? Some event in the brain, a vibration in the brain. Those are kind of our modern serious materialist views. And that did have some influence in Europe. So there is a little bit of a backwards influence where the Brits were reacting against the dualism of European philosophy. But some of their ideas moved back into Europe and inspired some French philosophers. And of course, remember, Descartes was French, and he was a dualist. And so in part, some of these newer French philosophers were also rebelling against the Cartesian mainstream here. So these guys were called sensationalists. It's a term that means a lot to us nowadays, but it just basically means here that they're putting all the emphasis on sensations, that's where the action is. To explain the mind, we need to understand what sensations are. Again, that's when the sensory receptor is activated by a stimulus. So when light activates the photoreceptors in the eyes, or when sound waves activate the hair cells in the ears, or when pressure at the skin activates the receptors in the skin. Those are all sensations, and this is where things begin. So everything begins with a sensation, and then this triggers an event in the nerves, which triggers an event in the brain, and all knowledge is built up out of those sensations. And that's, of course, an example of empiricism. It's an example of determinism, because again, we're determined by our sensations. It can be an example of reductionism, right? To explain away all the complexities of human mind and consciousness and the knowledge is just sensations. But it also motivates us to study the physical side of sensations, right? Which says if all knowledge is going to come from a sensation, and sensations occur at the level of these actual receptors in the eyes and the ears and the skin and the tongue and the nose for smelling and so forth, we need to study these sensory systems in terms of the anatomy and the functional anatomy and physiology of the sensory nerves. And that's a big thing. That's a big theme here, right? Understanding how these nerves work. How do these receptors work and how do their nerves work? There's very much a materialist monist kind of view here, right? To understand knowledge and consciousness, we just need to understand the nervous system, is what it boils down to. So clearly, this is going to begin neurophysiology and neuroscience. One of the first French physiologists to reject Descartes was Pierre Gassendi. And Gassendi has just one quote we want to focus on. He said, remember Descartes said, "I think, therefore I am." And by doing so, he's espousing this idea that this internal mental reality is really what we know, and that's all that we can say is real and so forth. And Gassendi says, no, right? It's not this internal mental reality that's important. It's just the fact that motions of the body and in the nervous system, that's what's going on here. That's the real essence of things. So it's not thinking that's important, it's moving. I move, therefore I am. Julian Lamatrie, writes the book Man on Machine, Man as a Machine, right? Is a machine. And so, again, he's kind of taking Cartesian ideas of this mechanical theory of the body and suggesting, of course, that everything that we think of about mind is really also explainable in terms of those simple mechanical principles. That whatever is going on in the nervous system in terms of those physiological states in the brain That's how we're going to be able to explain what we think of as mental states. They are really just mechanical, physical, physiological states. Condillac, interesting guy here. Condillac gives us the idea, that's not listed on the slide here. He asks us to imagine what he calls the sentient statue. So imagine you've got a statue that's made entirely of stone, a solid block of marble. But let's also pretend that we can stick a living brain into its head, assuming such a brain could actually be alive. It's there. And he says, would this brain be conscious? And his claim, Condillac's claim, is no. It would have no thought, no awareness, no consciousness. But if we are able to also then give this statue some eyes and a nerve that connects the eyes to the brain, this brain would now be conscious. And the question is, what would it be conscious of? Well, it would be conscious of whatever it sees, right? It receives visual sensations now that it has eyes and an optic nerve, so it would be conscious of visual sensations. If we gave it an ear and an auditory nerve, it would also then become conscious of auditory sensations and would also perhaps then form some associations or connections between visual and auditory things. And there would be a little bit more complexity to its knowledge because of that. If we gave it a nose and the cranial nerves that feed into for the sense of smell, then it would be conscious of olfactory sensations. And it would be able to form associations between olfactory sensations and the visual and the auditory ones, and those complex interconnections would add more complexity to this statue's cognition. And then we could give it a tongue for tasting and a skin for feeling, touching, and it would get more sensations and more complexity and more connections between everything. And this is where knowledge comes from. Knowledge ultimately is explainable by talking about those sensory inputs, right? And so this now lets us talk about comparative psychology, because what we're going to say is that if two animal species, say a human and, I don't know, a rabbit, a dog, have similar sensory organs, that is, they have compound eyes, they have skin for touching, noses for smelling, tongues for tasting, and ears for hearing, well, then their sensory inputs are the same, right? And if since all of that is the ultimate foundation of knowledge, then isn't the knowledge, the nature of experience or mind or consciousness, isn't it basically the same in other animals as it is in humans? So basically what we think of as psychology is the study of the psychological life here, mental life. It's something that we can do both with humans and with animals, right? And this justifies the study of what we would call animal psychology. And comparative here means comparing animal behavior to human behavior and suggesting the same underlying principles are at work in both cases. And so that's exactly what Condillac is getting at here. And this is in contrast to Descartes, because Descartes suggesting that the mind is somehow special, right? I think, therefore I am, and he equates it with the soul, but he says that it only exists in humans, right? Only humans, he believed, had a pineal gland where the soul would be, and that animals are much more mechanistic. Essentially what Condillac is suggesting is that we're just going to throw away the soul in the human side and treat human psychology as the more mechanical animal psychology that Descartes was already talking about, which is consistent with Lamettrie's idea that we are machines. And so that's it, right? So everything just is pure bottom up. Everything boils down to sensations. It's the same as Locke's theory of ideas, just without the dualism that Locke was talking about. So that's what the French sensationalists give us. They give us this philosophical idea that everything boils down to sensory input. So certainly physiologists of various sorts get to work on trying to understand the nature of how eyes work. The lenses, the cornea, the receptors, the nerve. How do ears work, right? Understanding the anatomy of the eardrum and the three bones and the cochlea and all that kind of stuff, and the nerves of the nose and the tongue and the skin. How does it all work? Another kind of theory to emerge in the early 1800s here, Mueller gives us what he calls the doctrine of specific nerve energies. He believes that he has discovered Kant's innate categories by understanding the nature of nerve impulses. He believes that the only reason we experience the world the way we do, say, visually, having visual sensations, is because we have an optic nerve, that the optic nerve itself imparts a peculiar quality to input to the brain, right? And so the optic nerve has its own specific energy. right? And that's what creates visual sensations. Likewise, the auditory nerve has a different energy that creates sound sensations. As an example of how the optic nerve is only really capable of responding with visual sensations, is it possible to stimulate the optic nerve without looking at something? Technically, we can. Normally, of course, if I look at something, light enters the eye activates the receptors, and the receptors ultimately synapse onto the optic nerve. But I can also poke my eye. I don't recommend you do this, but if I stick my finger here on the outside of my eye and press in on my eyeball through the eyelid, I will see over on the opposite side of my field of view, I will see little splotches of color and light just kind of flashing in my field of view there. Because what I'm doing is I'm actually pressing on the retina gently, but that pressure stimulates those cells, and the activity of those cells is registered by the optic nerve as a splotch of color and light. So therefore, no matter how the optic nerve is stimulated, its response is always some kind of splotch of color and light. So Mueller's idea here is that the reason we see color and brightness as properties of visual sensations is because that's a property of the optic nerve. So his claim would be is that if we had this ability to disconnect the optic nerve from the eye and plug it into the ears, that the sound waves that activate the mechanical parts of the ear would eventually then be stimulating the optic nerve, and we would end up seeing sound. And then correspondingly, we could take the auditory nerve and connect it to the eyes, and we would end up hearing light. Each nerve contributes its own energy to our experience and shapes our experience. And of course, this is what Kant was suggesting. It's consistent with Kant's idea that the brain is where the innate categories come from, and the brain imparts a structure onto our perceptual awareness. So in early neuroscience, now that we're into the early 1800s here, and people are going to want to start looking at the brain and trying to figure out what's going on in the brain, one of the themes is what we'll call localization of function. The idea here is that we want to pick out various parts of the nervous system and figure out what they do. Right? We can look, just with gross anatomy, you can look at parts of the nervous system that are easily identified to the naked eye. You can look at the cerebral cortices and all the different gyri there and say, what does this one do and what does that one do? You can look at subcortical structures like the thalamus and hypothalamus and the cerebellum and the brainstem and all of those parts and say, what do they do? So if we think that each identifiable part has its own function, then we say that those functions are localized to those areas, right? And that's one of the approaches here. One of the earliest approaches comes from this guy, Franz Gall, who is kind of problematic here. This is Gall's phrenological map. So phrenology was his idea here. So he's believing that localization of function is something that's a key element of how the brain works. Each area, each portion of the nervous system has its own particular function or job that it has to do. And when it comes to higher mental functions, that is the things that make us more human and intelligent, our language and our spatial abilities and our logical abilities and mathematical abilities and so forth, that these are all in the cortex as opposed to the more primitive brain areas. And one of the principles that Gall uses is that when it comes to talking about the brain, bigger is better, right? Which has actually been a problematic idea in the history of neuroscience, that bigger brains mean more intelligence. It works pretty well in making across-species comparisons. It does not work quite as well when making a within-species comparison, right? It doesn't mean that one person with a bigger brain than another means that big brain person is more intelligent than small brain person. Famously, people have Einstein's brain is on display at a university, I think, in Israel, and famously his brain is pretty much normal in all respects, right? So Gall says more is better, but it's not just about the overall size of the brain. For Gall, functions are compartmentalized, meaning that each little section of the cortex is a compartment, that is a closed-in, fixed, finite region, fixed boundary region that has a specific job. And that's what I have in this other slide of the phrenological map, is that each one of these little segments here with a number and some words in German there, that's where a specific mental ability is to be found in the brain. So if you have an advanced skill in one of these areas, let's just pick one of these, say this one right in the middle, and say that this is what Gall believed to be the logical thinking skill location in the brain. If you're really, really great at logical thinking, then this compartment here in your brain would be larger compared to somebody who was not a very logical thinker. But where does the extra cortex go? Because these boundaries are meant to be fixed. So therefore, this one area here cannot invade its neighbors. Right? Rather, it has to bulge out. Right? So it's going to bulge out, and that's going to cause a bump somewhere on your cranium. And so Gall, as we see in this other picture, is using a craniometer to measure the elevation of different points on your skull. So the idea is that this looks like it's drilling into the person's ears, but it's just clamped on there. And so this semi-halo is coming around, and then you move this stylus all the way around the circumference of the halo, and you can record the elevation of the stylus at different locations. And so you can figure out which areas are raised and which ones are depressed. And of course, you can rotate the halo all the way around the whole head and measure the elevations of all the different points. And you would then be able to figure out this particular person's particular abilities or capacities or aptitudes, because you would know which ones are elevated, and that's what they're good at, and which ones are depressed, and that's what they're not so good at. Now, there's not really much evidence to suggest that this is right? That not only is the bigger is better idea problematic in neuroscience, but this idea that bigger compartments correspond to better ability is, there's really no evidence to support that. But of course, another related question is, how did he generate this map in the first place? How is it that Gall came to believe that this is where logical thinking is, and maybe this over here is where musical thinking is, and so forth? Well, to some extent, it was based on case studies. That is, he might know somebody who was a great musical genius, virtuoso musician, and he would measure their skull and find the highest point on their skull, and then conclude, oh, well, that highest point must represent where their musical abilities are coming from. And so he would just assign that based on an N of 1 study. Some of them, the rest of them maybe, were complete fiction. Right? There was just basically zero evidence to support his claim that this was where this particular faculty resided in the brain. And of course, we also know in the end that the brain doesn't bulge out in different locations, causing the particular unique pattern of anyone's head shape. So pseudoscience is what we have here. But we have some other methodologies that begin to emerge, that some of them are not quite scientific in the sense that dealing with case studies is not always the most scientific thing in the world, but at least some applications of this can be used in a more scientific way. So the first method is the study of the method of ablation. So ablation means destruction of nervous tissue. In modern methods, we call it lesioning, which is much more precise, but in the 1800s it was much less precise. One of our first examples of it here is the story of Phineas Gage. So this is a case study, just an N of 1, right? So you may know the story. Phineas Gage was a foreman on a railroad, and you see him here with his tamping iron. This is a large metal rod that was used to tamp blasting powder down into a hole that was drilled, and the idea is they're blowing up, rocks and mountains to make way for the railroad. But as he was putting the tamping rod down into the hole with his head over it, looking down to make sure he's doing it, explosion goes off prematurely. It launches the rod up into his face and destroying his left eye. This photo is reversed, and we see researchers have in recent years taken his skull and done a nice MRI so they can actually trace the path that this rod took, so we can figure out exactly what part of his brain was destroyed. Now, he survived this, as you can tell in the photograph. He's still alive, sitting there holding his rod. So one of the benefits of the MRI is we were able to actually understand why he survived. This is usually a fatal injury for a lot of people to have a serious head injury like that, penetrating head trauma. the arteries of the brain actually grow in very specific locations, and there's not a lot of individual variability in where those arteries go. And the researchers were able to show with the MRI that that rod missed all of those arteries, even just by millimeters, but it missed them all. If it had nicked one of those arteries, he would have bled out. He would have had a cerebral hemorrhage and died very quickly. So that was the lucky part. Obviously, not very lucky in general to have this happen to him, but that's why he survived. But now we also can know that this part of the brain was destroyed, the prefrontal cortex, and there's many stories told about how his personality was altered, and that might be relevant. But more consistent is the fact that we learned that when he tried to go back to work, he could not really function on the job anymore, because in a managerial position, he was required to plan, problem solve, engage in decision making, and he had trouble with that. Right? He had trouble foreseeing the consequences of his actions, and that's where we think we learned a lot about the importance of the prefrontal cortex. Again, that's just a single case study, but using the ablation method, we see the discovery of, say, the Bell-McGindy law. So two different physiologists, Charles Bell and Francois McGindy, separately discovered this principle. They were both working with animals, I believe rabbits, at least in the case of Charles Bell, and one of the elements of the spinal anatomy is that if you look at the nerve roots that come off the spine, or the nerves themselves that come off the spine, they have two separate roots, right? So there's a root coming off the front of the spine called the ventral root, and then there's a root coming off the back side of the spine called the dorsal root. And nobody knew what the purpose of those two separate nerve roots were, right? What's the reason why each nerve has actually kind of a dual connection to the spinal cord? So Bell came up with the idea of snipping one and leaving the other intact, right? So let's take the nerve that innervates the arm, right, of a rabbit, let's say, and let's sever the dorsal root but keep the ventral root intact. In this case, what happens is that Bell observes that, and later McGindy the same thing, that the animal apparently has no sense of touch. That is, if you apply a painful stimulus to the limb, they don't react, but the animal is still capable of moving the limb. This kind of suggests that the dorsal root plays a role for bringing sensory input in. That's what we call the affective signal, whereas the ventral root contains the effective motor signal that controls the muscles. So, of course, we repeat the experiment on a different animal by severing the ventral root and keeping the dorsal root intact. And now what we see is that the animal cannot move the limb, but they do respond to a painful stimulus. So supports the idea that, again, ventral roots are the motor signal, and dorsal roots are the sensory signal. Later studies involved animal studies where they would just damage lobes of the brain, like damaging the occipital lobe of the brain and discovering how it affects vision, and damaging the temporal lobes and observing auditory impairments, or damaging the frontal lobe and other parts, not the prefrontal cortex, as we saw with Gage, but a little farther back, and that alters motor ability. Another method that emerged in the mid-1800s is the autopsy method. This helps us understand what different parts of the brain do, because what you do is you have a patient who has a neurological disorder. That is, they have some deficit in some ability. Now, they didn't have imaging studies, so that they couldn't X-ray or do a CT scan or MRI to see what's going wrong in their brain, but when that patient died, somebody could do an autopsy and find out what's going wrong in their brain, or what was going wrong in their brain. And the first case of this was done by a physician named Paul Brocaw, discovering now an area of the brain that is named after him, and correspondingly a speech disorder that is named after him. So Brocaw's patient was admitted to this facility with language disorders. This patient had a great deal of difficulty producing language. And over the years that this patient was in this hospital, his language declined very dramatically, to the point that while he was sort of near the end of his life, he could only say tan tan, and that was all that he could say. It was just very effortful and difficult to produce any language whatsoever. This is called an aphasia. When you have a neurological language disorder, that's an aphasia. In this case, it's an example of what we'll call a productive aphasia because he has difficulty producing language. But importantly, this patient could understand language. So if someone spoke to him, gave him a command, he could follow this command. So when the patient died, Brokaw does the autopsy, and Brokaw discovers lesions here in the frontal lobe of the brain, where I'm circling here, what we'll call Brokaw's area now, due to syphilis, right? So syphilis untreated ends up in later years becoming an active infection in the brain. It had actually spread into the neighboring motor cortex in the patient's later life, which explains why the patient also ended up having some right side body paralysis near the end of his life. But Brokaw was able to show that the lesions originated here in this particular gyrus of the frontal lobe, and that since the patient's earliest symptoms were speech production difficulties, then this is how we know what Brokaw's area is for, right? Brokaw's area is the speech production center of the brain, but not the speech comprehension center of the brain. That would be Wernicke's area, which was discovered a few years later over here in the temporal lobe on this particular gyrus. Wernicke had a patient who had kind of the opposite problem from Brokaw's patient. So Wernicke's patient could produce speech, but they could not understand speech. So this is a receptive aphasia. We call it Wernicke's aphasia now. Wernicke does the autopsy and discovers the lesions are in the temporal lobe, and we get Wernicke's area as the speech perception center of the brain. Here, in this image, we see a more modern theory of how language works, with perception occurring in auditory cortex and Wernicke's area, which is then transferred to Brokaw's area, where we formulate our responses, which communicates with the motor cortex, which actually controls our speech articulators. The angular gyrus and the visual cortex are used for reading, which feeds into Wernicke's area when we recognize words that we read. The third and final method emerging in the 1800s for the study of brain function is electrical stimulation. So, applying an electrical current to the brain, we can observe how it affects the response. Doing this with dogs, researchers are able to map out motor cortex, and we can see that there's basically a map of the body, and that isolating responses of these cells in different locations causes the animal to move different body parts, and they were able to then map out which body part corresponds to which part of the motor cortex. This is a cross-sectional view. If I go back to this previous slide and we look at the motor strip here, we can see that they would just move the electrode up and down and see what different body part moved. So it wasn't done with humans. It was done with animals initially, dogs primarily. In later years, there was some human research too. Here's a few other important discoveries about nerve function that emerged in the 1800s that are all worth mentioning. Helmholtz, as we previously covered, was doing some physiological research, and in fact he was the first to show that nerves conduct not instantaneously, but rather they take time. And so his own professor, Mueller, who was the one who gave us the doctrine of specific nerve energies, believed that nerve impulses were instantaneous, right? That the very moment a sensory impression is registered at a sensory surface, it's also immediately registered inside the brain. But Helmholtz showed that it does take time to conduct the impulse along the length of the nerve, and he developed reaction time studies to show this. So the basic idea is that he would put a stimulus on someone's foot, on their heel, just a small electric shock. And you're just supposed to wait till you feel the shock and you press a button, and he records your reaction time. And the idea is that your reaction time would be determined by the time it takes the signal to travel up your leg, up your spine, into the brain, and then for that signal to travel out of the brain, through the arm, to your finger to push the button. But then he moved the stimulus. So he moved it from the person's heel to their knee. And of course this makes the entire path a little bit shorter, because now it doesn't have to travel all the way up the leg. It only has to travel from the knee up instead of from the heel up. And what Helmholtz discovered is that the reaction times got faster. So the only way to explain why the reaction to a knee stimulus is faster than a heel stimulus is to explain it in terms of the shortened distance that the nerve impulse has to travel, meaning that the nerve impulse travels at a constant velocity, and when it has to cover a shorter distance, it leads to a faster response. And he was actually then able to even estimate the speed of that by looking at the time difference between the two responses. Golgi and Cahal, they both did some important stuff. So neurons are actually notoriously difficult to study using microscopes, and so of course we had to wait until we had good microscopes to be able to really see them. But microscopes had been around for a while, and still researchers had trouble studying nerves because when you look at cells under a microscope, you have to stain them, right, to give some contrast so you can see them. Neurons don't stain the same way other cells in the body stain, and so it was very difficult to start getting a good sense of what they were like. But Golgi invented a stain that worked. And then Cajal, Santiago Ramon y Cajal, he took the Golgi stain and he started using it to map out how neurons looked. And so this idea that we know what neurons look like, that we have the dendrites in the cell body and the axon and all the many interconnections and the web-like appearance of it, that is Cajal's discovery. In fact, he made these extraordinarily detailed drawings of neurons that were used, and so many are still used in medical textbooks today. So if you want, go on to Google and Google Santiago Ramon y Cajal, and you will see his drawings. Charles Sherrington studied reflexes because he realized that there were some reflexes that required a synapse in the spine, and he was able to understand how the synapse works as a sensory input into the spine, a spinal reflex, and a motor neuron that sends a signal back. And he was also able to reveal some aspects of the difference between an excitatory synapse and an inhibitory synapse. Galvani was the one who discovered that this entire synaptic activity is somehow electrical in nature. So what he had done, this is an example of a serendipity, just an accident. He had a piece of frog leg muscle that had a nerve running through it in a petri dish full of saline solution. And he accidentally dropped a live electrical lead into that dish. And when he did that, he noticed that the muscle twitched. And he thought, that's interesting. Is there something about this muscle and the nerve that reacts to electricity? So he takes the muscle out and he puts a positive lead on one end and a negative lead on the other end, and he passes a current through the nerve. And of course, every time he passes a current through the nerve, the muscle contracts. So he realizes, oh yeah, this signal is electrical. That's what's going on. The nerves conduct electricity. It's actually around this time then that Mary Shelley wrote Frankenstein, right around this understanding that the nervous system is somehow functioning with electricity. And then later on, Otto Lowe, we showed that it wasn't purely just electricity, right, but it was actually a chemical called a neurotransmitter. And Lowe, we discovered the very first of the neurotransmitters that we learned about, called acetylcholine. Now what he was doing is he was looking at a frog heart. So he has a frog heart floating in a dish of a beaker of pure water, just distilled water. So there's nothing else in there except H2O. It has a nerve attached to it, and so Lowe would stimulate the nerve with electrical inputs, and this would cause the frog heart to beat. Then what Lowe did is he took this liquid that heart had been floating in, which was pure H2O, and he transferred it to another beaker that had a different frog heart in it, and lo and behold, this new liquid causes that other frog heart to start beating. So what he's concluding here is that when the nerve is stimulated and the heart starts to beat, this causes the production of a chemical, right? And it's this chemical that actually triggers the process. And so once we transfer the chemical to a different frog heart and the other frog heart starts to beat, it's because the chemical is really the agent of action here, not the electricity. Let's take a look at another major sort of early neuroscience and experimental psychology. And this is the one that looks a little bit more like experimental psychology and less like neuroscience. And this field is called psychophysics. You can break that word down. Psycho for psychological, physics for physical, and it's kind of a mapping from psychological to physical, or from physics to psychology. Part of the goal here was to create a mathematical psychology. So being able to describe sensory responses mathematically was seen as something that would be good scientifically, objectively. If we think about positivism, when we think about objective observation and quantifying things, psychophysics certainly had that as a goal. And of course, what this also means is that we would ultimately, following the sensationalist approach, be able to explain what we think of as mind as really just sensory issues, right? It's the study of sensations. We want to study sensations, not just at the level of the sensory anatomy, right, of the organs, but also just of what's the nature of an internal sensation itself. And this is going to take us to Herbart. So the way we study sensations is to study thresholds. And Herbart is the one who gives us the first idea that there could be such a thing as a sensory threshold. He calls it the absolute threshold, or the limit of consciousness, the alignment of consciousness. We think about, go back to chapter 2, we were talking about Zeno's millet seed example, where he said a single seed hits the ground and it makes no sound, so therefore it doesn't make sense that many seeds make a sound. But Zeno didn't know about thresholds, and so it's not true that a single seed makes no sound, but rather it makes a very weak sound that, when combined with many seeds, is multiplied to become something large enough to cross the threshold. And Herbart realizes that that's the explanation. And he says that there are some sensations that are strong enough to cross the threshold and enter into the conscious mind, and then there are others that are not. So the absolute threshold is considered to be the weakest stimulus that we can detect, that is, that we are conscious of. There's another kind of threshold, we'll call it the difference threshold. So the idea is that if a sensation changes, how much does it have to change before we notice the change? Right? So that's a difference threshold. Originally it was called a just noticeable difference, J and D. So that just means, again, what difference is the one that is just noticeable? And so psychophysics as a field is a method for studying absolute thresholds and difference thresholds. How can we quantify thresholds? So let's first talk about the methods themselves. How do we measure a threshold? How do we figure out what is someone's actual absolute threshold for, say, sound, right? If you've ever taken a hearing test, you are basically experiencing psychophysical testing. They're trying to measure your auditory thresholds, the weakest sounds that you can hear. One way to do this is with the method of limits. The method of limits was the first one that was invented. So we would basically say, let's turn the volume of the sound all the way down to 0. And let's turn it up a tiny amount and ask our participant, if they can hear it. They're going to say no. So I'm going to turn it up another tiny little increment, and they still say no. Now turn it up again, they say no. I turn it up again, they say no. And then I turn it up one more time, and now they say yes, I can hear a tone. So the idea here is that I've presumably found their threshold, because I just increased from 0, and I kept going up and up and up until now they say they can hear it. Right? That's the weakest sound that they can hear. That's called an ascending trial, because I'm increasing the intensity until someone says yes, they can hear it. But we can also do a descending trial. What if I turn the volume up moderately loud, and so they say, yeah, I can hear it. I turn it down a little. They say, I can still hear it. I turn it down a little. I can still hear it. Turn it down some more. I can still hear it. Turn it down again. Oh, it's gone now. So now I found the point, again, that represents kind of not the weakest sound they can hear, but maybe the loudest sound they cannot hear, right? That's called a descending trial. If an absolute threshold is really a fixed kind of thing, you would expect to get the same results in each case, right? That the actual intensity that you've settled on here is the same. But as it turns out, that's not true. You get different results, sometimes wildly, drastically different results when you do an ascending trial versus a descending trial. How do we explain this? Well, there could be various explanations for it, biases and so forth. But maybe what we want to do is improve our method a little bit. And so this is where the method of adjustment comes in. So now the idea is that it's the subject themselves, that's what the S stands for there, the subject can actually control the volume knob themselves. So they get to turn the knob back and forth, and they get to set the knob right at their own threshold. And because they're kind of going above and below, they don't adapt, and they don't have any of these biases that might influence their judgments in using the method of limit. So it's seen as an improvement. Even better is called the method of constant stimuli. This is where you randomly present different stimuli, so that way the subject never has a chance to have an expectation or a bias to know if the sound is coming or not. So if you've ever had a hearing test, as I said before, you would have experienced the method of constant stimuli. Usually they would present the tones in random order to prevent you from guessing. This first slide here, under psychophysical laws, represents the first initial research in psychophysics, and it was done by Ernst Weber. And so it gives us one of our first laws in psychology, Weber's law. And he was studying J and D's, just noticeable differences, or difference thresholds. So again, he wants to know what's the amount of change required to produce a noticeable change. Before I explain that formula below, let's use an example. Grab my book here. He was studying touch-related stuff and perception of weight. So I hold the book in my hand, and I can talk about how much it weighs, right? And if I were to add some weight to it, I can add my pen to it, right? And I could then ask, does it feel heavier? And the answer might be no, right? Instead of a pen, what if I just added a grain of sand to it, right? And obviously I would say no, it doesn't feel any heavier with just one little grain of sand. But maybe I add another one, and then another, and then another, and then another. And the question is, how much weight do I have to add before now I say, okay, yeah, that feels heavier now. I definitely can feel that, right? That would be the J and D, or the difference threshold. And so Weber's law, what he's saying here is that if R represents the initial weight of the book, delta R represents the amount of change, right? Delta in math is a change. Delta R represents the amount of change in R, that is how much has to be added to it to create the J and D. And the ratio of those two things should be a constant K, right? So let's pretend the book weighs 1 pound, which it doesn't, but let's pretend it weighs 1 pound. If I had to increase it by 0.1 pounds, right, that's a 10% increase. So that would be our K, right, 10%. If I started with a different book, a bigger book that weighs 2 pounds, the idea is that I would have to increase it not by 0.1, but rather by 10%, which in this case would be 0.2, right? So suggesting that the J and D is always the same constant proportional increase, whether it's 10% or whatever it happens to be for a particular sensory system that you're studying, you would expect to see that same constant hold true. And that's Weber's law. K is referred to as Weber's constant. This is a pretty useful principle that predicts a linear relationship between the stimulus and the response. But as it turns out, Weber's law doesn't actually hold. It only holds if you're studying certain stimuli and only in sort of medium intensity values. But if you get into the really loud sounds or the really heavy stimuli, it turns out nonlinear. That is, K starts to change, right? K would represent the slope of a line rating delta in terms of graphing delta R as a function of R. And so K is obviously not constant. And this is where Weber's student comes in, Fechner, who fixes the problem here. So Fechner says, oh, it's a logarithmic function. The idea is that we have to compress the scale. So he replaces the delta R with S as a unit of sensation. And he considers that the sensory scale is linear. It's built out of equally sized units. But the physical scale R, that is represents the actual physical intensities of the stimuli, that follows a power scale, right? An exponential scale. And in order to make an exponential scale and a linear scale match each other, you have to use the logarithm, right? The logarithm is basically taking the root of the exponent of that scale and compressing it so that the two scales match. And then you can recover the linear function with K as its constant here. So that's what Fechner's law is. It's basically the same as Weber's law, right? S is what delta R used to be, which is a sensory response, as opposed to the stimulus intensity R. And they are related to each other through a logarithmic function with the slope of K, which is again Weber's constant. Even better is jumping way into the 1900s, actually, and as far as the 1950s, we get Stevens' power law. So Stevens recognized that the logarithm has limited utility for mathematically modeling these different, what we're calling psychophysical functions, that is, being able to map the stimulus energy to a sensory response. But if you use a power law, that is, instead of a logarithm, you take R to some power of n, you will get different kinds of curves, right? With the idea that, if it is a straight line, you'll have n of 1. If it's certain other curves might have an n of 2, or you might get a square root function, an n of 3, or whatever, quadratics. So all these different curves can be modeled using a power law. And so that's really the lesson of Stevens' power law here, is that with these different functions and the different exponents, you will be able to improve your modeling abilities. And if that's ultimately the goal, is to improve the ability to successfully mathematically describe the relationship between a physical stimulus and a sensory response. Stephen's power law gives us the mathematical power that we need to do that. And so these are psychophysical laws that, as a study of sensory systems, might be thought of as a form of neuroscience, but at the same time, these kinds of experiments actually involve people to provide verbal responses in terms of saying, yes, I can hear it, no, I can't, right? And so this involves more psychological stuff as well. So this is some of the obvious beginnings of experimental psychology, especially with Weber and Fechner there. And thank you, that is the end of Chapter 7.